Good morning, everyone. So the topic I'll be talking about today is a method that was developed in our group fairly recently, and it concerns the synthesis of potentially functionalized nitrogen cycles, and particularly piperidines. Cyclic molecules that contain a nitrogen atom are useful in various fields, and particularly in pharmaceuticals and agrochemicals. For example, if you have a look at the Food and Administration database, you'll find that more or less 2/3 of the drugs that have been approved possess a nitrogen heterocycle, and the piperidine core is the most frequently used ring system. Additionally, molecules that are rich in sp3 carbon atoms, particularly sp3-rich heterocycles, gain a lot of interest in medicinal chemistry. And the reason why is that they have been able to show that the fraction of sp3 hybridized carbon atoms in drug candidates is directly correlated to important physicochemical properties. For example, increasing the sp3 character of a drug resulted in higher aqueous solubility and lower melting point. They've also found that sp3-rich compounds are more likely to succeed in clinical trials, and that the presence of chiral carbon was also an important factor in the clinical progression of a drug-like molecule. So in this context, in the Hoteite group, we have been working hard to access these important building blocks. So the initial idea that emerged in our group was to develop a formal 3+3 strategy towards piperidine, where you'd be able to use some kind of 3-carbon-containing zwitterion equivalent that would engage in an addition and cyclization reaction with strained 3-membered ring systems, such as a azorhidine or an epoxide. So the first zwitterion that was found was a palladium-stabilized trimethylene methane that actually did the job very well and gave the corresponding piperidines in good yield. Then they moved on and devised a main group version of this reaction, and finally, again using main group organometallics, they were able to generate 6-membered rings with different functionality by using different kinds of 3-carbon-containing zwitterion. And finally, in order to showcase the diversity and efficiency of this chemistry, they applied it to more complex targets such as natural products. So one of the good things about this chemistry is that it allows you to make a range of nitrogen and oxygen containing heterocycles. However, it would be more useful if you can do it with incorporation of functionality. And in addition to that, this chemistry is stereospecific. So what I mean here is that if you want to make an enantiomerically pure 6-membered ring product, you have to start with an enantiomerically pure 3-membered ring. You cannot control the stereochemistry in the 3+3 reaction itself. Therefore, the initial idea of a 3+3 reaction evolved into a 4+2 version of this chemistry. And to do that, what they essentially did is to homologate the 3-carbon-containing zwitterion to a 5-atom-containing dipolar reagent, which contains the nitrogen atom. And so here the idea was to make an enolate, which would react with the cation part, and then this nitrogen cyclizes and condenses around to generate a 6-membered ring. So the key thing over here is that a new stereogenic center is created, so it allows us to develop an asymmetric version of this reaction. And another thing that is nice is that you can see that these scaffolds have lots of functionalities. So there's also the opportunity as well to take these products in all sorts of different directions towards exciting targets. So, for example, taking something like this cyclic carbamate and a palladium-catalysed, you have oxidative addition into the allylic carbon-oxygen bond, gets us over here, and then loss of carbon dioxide would give us this amide, which could deprotonate an alpha of the carbonyl to form the enolate. And then these two come together to generate this allylation product. And finally, if you can engage this compound in a condensation reaction, then we're there. So we were able to do that. So taking this book substituted cyclic carbamate in the presence of a ketone, we could indeed get to this allylated product, and then simply throwing TFA in the same pot. You don't have to purify the intermediate. It removes the book group and promotes a cyclic condensation. So here's just 3 examples from this initial study, just to give you an idea of scope. So for example, taking a simple 1,3-diketone, again with palladium-catalysed, and then throwing TFA in the same pot, it gives this functionalized single piperidine ring. If you then take a lactone with a ketone, substitute alpha to a carbonyl, you can generate a spirocyclic piperidine, And then if you take on the opposite, you have a cyclic ketone with an ester outside the ring, then you generate a bicyclic piperidine. And I think this really exemplifies one of the nice features of this chemistry. Just by choosing, for example, whether you have the ester in the ring or outside the ring, you can dictate the type of architecture that you get in the product, spirocyclic or bicyclic. So just by choosing the type of substitution you have, you can make all sorts of different kinds of rings with various architectures. Fairly recently, our group has been engaged in trying to apply this chemistry in target synthesis. So we've considered 4 different classes of targets, 3-fluoro and 3-trifluoromethylthio-ketones, fused polycyclic piperidines, and 3-amino piperidines. So during my PhD, I was involved in the application of this chemistry toward the synthesis of the first 3 heterocyclic targets, and that's what I'll be talking about in the next slides. So it's the 3-fluoro and 3-trifluoromethylthio-ketones I'm going to start with. So these compounds have emerged as very important and are actually investigated in medicinal chemistry. And the reason for that is twofold. One fairly well-established thing is that when you put a fluorine atom into a molecule, particularly at a metabolically labile site, then you tend to reduce the metabolism because you have often replaced a carbon-hydrogen bond for a very strong carbon-fluorine bond. So you can enhance metabolic stability. But in the context of piperidines, putting a fluorine atom in the 3 position also reduces basicity enough to reduce hERG liability. And hERG is a very important anti-target in medicinal chemistry. Now, putting the fluorine in the 3 position is key because if you put the fluorine in the 2 position, it's chemically unstable. The nitrogen will kick off the fluorine and your compound will decompose. And if you put it in the 4 position, it's too far away to attenuate the basicity enough. So the 3 position is optimal. And there's a number of bioactive compounds that have emerged and have exactly this fragment in there. So there are several approaches that people have taken to make these compounds. The first one involves an electrophilic fluorination of piperidone. However, this approach suffers from lack of regioselectivity when non-symmetrical substrates are used. Another way is via the deoxyfluorination of 3-hydroxypiperidine derivatives. However, this reaction requires pre-functional substrates and exhibits low atom economy as well. Another common way to make these compounds is the aminofluorination of alkene, but this reaction requires to use stoichiometric amounts of toxic and expensive reagents. And more recently, the group of Glorious has reported a 1-pot deramatization hydrogenation of fluoropyridines, which gives a range of 3 fluoropyridines. However, the reaction requires high pressure in hydrogen, which decreases its operational simplicity. Whereas with our chemistry, we can access the substrates that we need to make the piperidines by allylation-condensation of an α-fluoro-carboxylic acid derivatives, and these are abundant and are actually very inexpensive. So our strategy potentially allows us to engage much cheaper sources of fluoride. So the methodology works very nicely. If you take a range of these compounds with the cyclic nitrogen book protected carbamate, you generate this compound which after GFAD protection gives this cyclic product. So we can make a range of examples where we have an aryl group in this 2 position, and we can also include a range of alkyl groups. We have been able to scale this chemistry up. So in one example, we've run the reaction with 3 grams of substrate, and we were able to isolate the product in 91% yield over 2 steps. So this chemistry is quite scalable. However, in order for this product to be used in other chemistries, it requires to be able to do chemoselective functionalization. And we've been able to do that. So here's some few examples of what we've done in our group. So we can do cross metathesis, so that allows us to react the alkene in the presence of other functional groups. We can carry reductive amination with triazoxyborohydride without affecting the ester or the alkene. And we can use lithium aluminum hydride, which quite remarkably reduces the ester without touching the amine or the alkene. What we can also do is hydrolyze the ester, as that promotes the decarboxylation, and so we can generate this compound, which could then be reduced to form this piperidine with good control of the ester selectivity. So we can carry out chemoselective functionalization of these products toward all sorts of different new starting materials. This chemistry proved to be successful in accessing 3 fluoropiperidines. We thought that it might be adapted to allow the incorporation of trifluoromethylthio groups. Due to its electron withdrawing nature and high lipophilicity, the SCF3 group can significantly modulate the pharmacological properties of bioactive compounds. However, to the best of our knowledge, only 2 examples of 3-trifluoromethylthio substituted piperidine have been reported. And therefore, our palladium-catalysed allylation-condensation methodology would be of significant value to the pharmaceutical and agrochemical sectors, and would also stimulate developments in the field of fluorinated nitrogen cycle synthesis. So then we examined the palladium-catalysed 4+2 annulation reaction with α-fluoro-β-ketoesters substituted ketones. And as you can see in the scheme, all substituted ketones proved to be excellent substrates for this transformation. And so we were able to make a range of 3 SCF3 substitute piperidines under mild conditions. However, unfortunately, these products proved to be unstable to chromatography, and so we used a borohydride reduction step prior to isolation. All these 2 3-trifluoromethylthio piperidines were isolated in good yields over 3 steps and with very high cis steric control. The X-ray crystal structure analysis of these 2 substituted products confirmed the relative stereochemistry of the major diastereomer, and the stereochemistry of all the other substitute products was assigned by inference. Finally, using the alpha-CF3 propionine, we were able to form a more substituted analog, and we were also able to make the 2 alkyl piperidine derivatives, although in low yield. Okay, it's not all great. One of the things that we really struggle with is to try to make this chemistry asymmetric. And actually, we have worked incredibly hard to try and improve the enantioselectivities, but nothing really worked. Either we obtained poor enantiomeric ratios, or we just got back a racemic mixture. So I'm not going to enter in details here about all the different approaches that we've attempted to get around this. However, here's a brief summary about some of the strategies that we've tried. So, despite having screened a large number of chiral, phosphine, and phosphoramidite ligands, we were not able to obtain any enantiomeric excesses over 10%. We also tried to induce asymmetry by attempting to form a chiral annulate via coordination to a chiral Lewis or Brunstad complex. However, this failed to deliver any promising selectivities. We've also investigated to use chiral auxiliaries, such as the methyl ester or Evans-oxaldenone. However, in the first case, we obtained a 1-to-1 diastereomeric ratio, and for Evans auxiliary, no reaction occurred, and we recovered back our starting material. We've also attempted to use a chiral hydrogen bonding catalyst, and in this case, the palladium-catalyzed reaction worked. However, the product was obtained as a racemate. And finally, we also tried to do a dynamic kinetic resolution on this substrate over here, as well as an enzymatic resolution of the lactam. But unfortunately, none of these strategies delivered any enantioselectivities. Okay, for the last few minutes, I'd like to talk about the other target I've been working on, which is the formation of fused polycyclic piperidines. So we thought that this palladium-catalyzed reaction would also be useful in order to access more complex polycyclic piperidines. So we decided to apply this methodology to a range of tetralose and indenone-based substrates. And as you can see, this chemistry worked quite nicely, giving the desired allylated compounds in high yields. So in a similar way as I mentioned earlier, if you throw TFA, it will deprotect the book group, and then the amine cyclizes to get these fused polycyclic heterocycles. Similar to the other type of product, these scaffolds contain orthogonal functionalities. So it also allows us to take these products and derivatize them in all sorts of different directions. Okay, so we were able to functionalize the tetravalent-based products further by doing reductive elimination to get to the saturated piperidine ring system. However, with the N-Heterocycles cyclic amines, we struggle to do that, despite trying hard by using different reduction conditions. So we've hypothesized that the issue arising here in this simple reduction step is maybe due to the formation of an unstable strain trans 5,6 ring system that would be imposed by the attack of the hydride from the opposite side to the aryl group. Again, I'd like to emphasize that this was our hypothesis. We cannot say whether the product is unstable and decomposes, or whether the transition state is too high in energy, and under these conditions, the target material decomposes. So, based on what I said earlier, reasons that reduction of the iminium salt by a radical pathway might ease the ring strain by the formation of a free radical that could interconvert to the thermodynamically more stable conformation. Therefore, we subjected the iminium salts to a visible light photoredox reduction that was recently reported by Wenger and co-workers. So, using ruthenium BP as a photocatalyst in the presence of ethyl and sodium ascorbate in methanol, we were indeed able to isolate the desired tricyclic piperidines as single diastereomers and in good yields. So the product stereochemistry was assigned as cis based on a positive NOE correlation between the hydrogen of the CH adjacent to the nitrogen atom and the aromatic proton. As we've successfully synthesized a range of polycyclic piperidine that contain orthogonal functionality, we then turned our attention toward product functionalization. We managed to do an oxidative cleavage of the exoalkene using ruthenium trichloride and sodium peroxide to form the ketone. And actually, this compound resembles an intermediate that was converted into horamine A by the group of Furstner. Additionally, we were able to derivatize the amine tetravalent-based product with allylated ethyl aluminium to give this homoallylic amine product in good yield and as a single diastereomer. And as you can see, this compound contains now a new synthetic handle that could be useful for further transformation. So, in summary, we have developed a robust method that allows the formation of a range of highly functionalized 3-fluoro and 3-trifluoromethylpiperidines. I showed that these products could be derivatized chemo and diester selectively to give novel and interesting fluorinated heterocyclic compounds. I've also showed that this methodology could be applied to indenone and tetravalent-based substrates to give highly functionalized polycyclic piperidines. Unfortunately, until now, a main limitation of this palladium-catalysed process is the non-selectivity. However, this is still an ongoing challenge in our group, and hopefully we'll be able to develop an asymmetric version of this chemistry. Now I'd like to end this presentation by thanking my supervisors, Professor Joe Hoteite and Professor Enrique Gomez, all the Hoteite group members, past and present, particularly Ben Allen, Chris Lakeland, and Victor Vasquez, who've been working on this project as well, all the technical staff, the EU for funding, and finally all of you who took the time and listened to this online presentation. Thank you.